Good evening, one and all. My name is Ubaid Tariq. I'm a PhD scholar at BSB Department of IIT Kanpur, and I am taking this session of uh, which is the problem-solving session of the course Cell Culture Technologies, which is taught by Professor Manik Das. So every week at 7 p.m. I take this live session, and all of you are invited to join the session. We will directly start with the questions that are given in the assignment of the week 4. The first question is poly D lysine contains dash number of nitrogen atoms. The answers are A 1, B 2, C 3, and D 4. The answer of this question is 2. The poly D lysine contains 2 nitrogen atoms. This poly D lysine, it's a biocompatible anionic polymer, and it comes with both two isoforms. One is the L isoform and another is the D isoform. And and the L isoform is found to be very susceptible to the enzymes, and these enzymes digest the L isoform. Whereas the D isoform is stable to digestible enzymes or degradable enzymes. So this D isoform, that is poly-D-lysine, it is commonly used to coat tissue culture plates or tissue culture flasks, and it is coated to enhance the attachment of the cells or to improve the adherence of the cells to the tissue culture plates. Actually, this poly-lysine, it is a it's a amino acid. It's a group of amino acids, and under physiological conditions, these poly lysines, they become protonated under the physiological conditions. And there are two nitrogen atoms, as you see in this schematic, in this picture. There are two nitrogen atoms in the poly lysine, and under physiological conditions, these nitrogen atoms or these nitrogen groups, they become protonated, and after when they become protonated, they attract negatively charged biomolecules such as DNA cells. As cells also have the net negative charge, these these cells and DNA, they get attracted by poly-D lysine, and these cells adhere to the culture plate of the cells. Usually, two types of materials are used as cell culture plates. Earlier, the glass were used, for example. Now, also the glass are used, for example, these glass slides or cover slips are made of glass, and sometimes cells are seated in that glass. And usually, the glass contains the negative charge, and it is coated with a polylysine, which contains positive charge, and after that, the cells also has the net negative charge. So the cells and biomolecules, they adhere to the polylysine. Nowadays, majority of the culture flasks are made up of polystyrene. These polystyrenic plates or polystyrene plates, they also have the net negative charge, and they are usually coated by the polylysine. Earlier, these flasks or glass plates, they were coated with the natural extracellular matrix components like collagen, fibronectin. But now that has been completely replaced by the coating of polylysine, because polylysine is a non-specific adherent which non-specifically adheres to all types of cells, because it only works by the mechanism of this polarity. It is a positive charge, and the, the macromolecules or cells which have a negative charge, they adhere to polylysine. Now we will move to the next question. I hope you have understood this. Next question is, which of the following is a synthetic extracellular matrix? The answers are A. Madrigal, B. Collagen, C. Laminin, D. Poly-L-ornithine. The answer is very simple. You may be knowing that others are the components of the extracellular matrix, and madrigal is also the ECM, as uh, I have told you about the madrigal in the earlier conversation. So the answer is poly-L-ornithine. So poly-L-ornithine, it is the synthetic amino acid chain, and is a positively charged or cationic amino acid chain, and it enhances the cell attachment. Actually, this poly-L-ornithine, it provides the biophysical and biochemical cues which are similar to the extracellular matrix. And this poly-L-ornithine has been widely used for coating the other plates for enhancing the attachment of the cells. So it's a synthetic extracellular matrix, and it mimics the properties of extracellular matrix, like some biochemical and biophysical property. It mimics these properties of the extracellular matrix. And this poly-L-ornithine, it has also been used with and it has also been conjugated with other molecules like laminin, ivermectin, and it is used for neural cell differentiation or neural cell attachment. Talking about extracellular matrix, so extracellular matrix is a major component of the tissue, as it is a major component and it adheres the cells. It adheres the adjacent cells. It also provides the biochemical environment, biophysical and physiological environment to the cells for the proper functioning of the cells. This extracellular matrix, it also regulates the tissue homeostasis, as it excretes various factors which help in the homeostasis of the cells. This extracellular matrix is also used to is also known to regulate the pathophysiological conditions also. In pathophysiological conditions, like in case of myocardial infarction, the extracellular matrix, which the extracellular matrix concentration is increased, so that heart, the wall of the heart gets the thickness of the wall gets increased and it becomes durable. So in pathophysiological conditions also, this extracellular matrix regulates these pathophysiological conditions. But there are some limitations of natural extracellular matrix, like every time we have to extract from the natural source. So there is a need to develop the synthetic extracellular molecules. So these are the building blocks extracellular matrix. So there is a need to develop the synthetic components of these. So for creation of the unique synthetic extracellular matrix or ECM, different types of materials have been used for their synthesis. Like I have said in earlier, poly-L or anything is one of the component. And the main purpose of these synthetic ECM is to use as they can be used as a engineered scaffolds in case of tissue engineering, as extracellular matrix is a component of the tissue, and and cells remain adherent to extracellular matrix, and these extracellular matrix and these can be used in the scaffolds so that the cells will get the natural biophysical and biochemical cues, and cell will grow properly in the ECM. And in case of this extracellular matrix also has an importance in case of 3D printing or 3D bioprinting. In case of 3D bioprinting, the cells are printed to make some organelle or organ. So extracellular matrix is important. So the cells can be embedded between the extracellular matrix in a mesh type of extracellular matrix. So there is a need to develop the synthetic extracellular matrix for these tissue engineering applications. Next question is, gelatin is a hydrolyzed form of. The answers are A. Collagen, B. Elastin, C. Laminin, and D. Fibronectin. What is the answer? So the answer is collagen. Actually, this collagen is a protein. It is present in the mainly in the connective tissue, and and upon controlled hydrolysis, this leads to the formation of gelatin. When the upon the controlled hydrolysis, there is the formation of gelatin, and gelatin also has different properties from the collagen. So it should be controlled hydrolysis. If it is uncontrolled hydrolysis or it is not controlled, then this gelatin can hydrolyze further and it can form the hydrolyzed collagen. You may be aware that there are two types of gelatin. There is a gelatin A and gelatin B. If the collagen is hydrolyzed by the acids, it's called acid hydrolysis. Gelatin A is formed. If it is if the collagen is hydrolyzed by the base, so it's called base hydrolysis, then the gelatin D is formed. Ultimately, this gelatin is also the protein is derived from the collagen by controlled hydrolysis. And this gelatin has a very important place in tissue engineering and 3D printing. And as a lot of scaffolds have been made using this gelatin as a scaffold or extracellular matrix. They are used as a scaffold for tissue engineering. And this gelatin has also the property that it gels on cooling it forms a gel. So it is widely used in tissue engineering applications. Another question is, atomic force microscopy helps us to analyze. The answers are chemical constituents in the bulk of material, B. Topography of the surface, C. Concentration of material, D. Electrical stimulation of cells. So, what is the answer? The answer is topography of the surface. So, atomic force microscopy, it's also called as scanning force microscopy or scanning probe microscopy. So, this atomic force microscopy, it actually it has a probe or which is called cantilever, and the sample is placed on the atomic force microscopy stage, and this cantilever has a tip which is very sharp tip, and this this tip it will be in contact with the sample. But there are two types. One is the contact AFM, another is the non-contact AFM. But most of the cases, contact AFM is used. And this, upon touching this sample, uh, the analysis is done by like simply the feeling or touching. For example, if you are touching the sample, sometimes you close your eyes and you touch some surface, and by touching you can feel the surface and you can tell what is the property, what are the properties of the surface, or how is the morphology of the surface. Like I said, it is the topography of the surface. So This tells the topography of the surface. Has a resolution of few nanometers, and uh, and this resolution is 1,000 times better than optical differentiation microscope. So, what happens when this sample, when it is placed in the surface or it's moved on the sample, when there are the deformation in the surface, there is a movement in this cantilever, and when there is a movement in cantilever, these lasers they are irradiated on the cantilever and they are detected by some detectors. When there is a movement in the cantilever, so these, there is also, because of that movement, there is a deflection in this laser light. There is a deflection in this laser light, and due to that deflection, which is measured, the deflection is measured, and overall the surface topography is measured. So it's simply like feeling the surface by your hand. Here they are using the lasers to evaluate it, how the surface is. So in this atomic force microscopy, the force between the sample and the probe is important, and the force is measured. That's why it's called atomic force microscopy. But there is no atomic part in this, as it is only the analysis of the surface, analysis of the topography of the surface is done by AFM. So it measures the force between the sample and the probe as a function of this. So it studies the Young's modulus of the surface or different properties of the surface like morphology, Young's modulus, how the surface is stiff. And there are two types of AFMs. One is the contact AFM, another is the contactless AFM. So in case of contact AFM, which also in case of contact AFM, the sample or the strip it is in contact with the sample, and it is moved in the sample. It's moved from one place to another place. So that to detect the morphology or topography of the surface at each point. So in contact mode also there are two types. One is the static mode or drag mode. In the static mode, the sample is at a static position. It is, for example, you are seeing the tip. It's placed in a static manner, like it's placed on the sample and then it is dragged from one portion to another portion. It's called static mode. Another is a dynamic mode or tapping mode. Tapping, as you as from the word tapping, you can relate to it. Like the here the it's tapped like this. This tip is tapped on the sample and it is simultaneously tapped and moved from one place to another place. So the deflection is measured. And another non-contact AFM microscopy in which this probe it is not in the contact with the sample. It is only 1 nanometer apart from 1 to 1 nanometer above from the surface. So this was all about atomic force microscopy. Now I will move to the next question. So next question is X-ray photoelectron spectroscopy is used to analyze. First answer is chemical constituents of the surface of material. Second is the topography of the surface. Third is the chemical constituents of bulk of material. D part or D answer is the topography of the bulk. So what is the answer of this question? The answer is A part, chemical constituents of the surface material. So in the next slide I will explain this. So this atomic X-ray photoelectron spectroscopy, or it's also called XPS, X-ray photon electron microscopy. So it's a surface sensitive quantitative microscopy. It helps to evaluate it is a quantitative thing. It's not only the quantitative. It gives the quantitative composition of the surface. So it's also the surface analysis tool or surface analysis spectroscopy, and it helps by the help of this XPS. The X, the depth up to the depth of 10 nanometer, the surface can be analyzed. So this X-ray or XPS, X-ray photon electron microscopy, it has a the sample is placed on the sample holder, and the focused X-ray beams are irradiated on the sample. After irradiation of this X-ray beams on the sample, so the photoelectric effect occurs, and because of these uh, X-ray beams, emission of the electrons from the sample occurs. And when these X-rays hit the surface or hit the material, these electrons are also called photoelectrons or photo-emitted electrons, and they escape from the top of the surface as these X-rays can only go up to the depth of 10 nanometer. And this after irradiation of this X-ray beam, the electrons or uh, electrons come out of the surface, and they are analyzed by the semicircular electron analyzer. And because of uh, and this and how now you can say that you electrons are irradiated. My electrons are coming out of the surface now. How the surface composition is detected by this XPS. So after these electrons are come, they are they come from the sample. Now they are analyzed by the analyzer. This analyzer analyzes the kinetic energy and the number of electrons. So it helps to detect the elemental composition of the surface. So the answer here was the chemical constituents of the surface material or elemental composition of the surface. And this XPS it also has a property that it not only tells the elemental composition of the surface, but it also tells the chemical state of the elements or atoms that are present on the surface. Overall electrical structure, chemistry of electrons, all these can be analyzed by XPS. The next question is contact angle on the surface is indicative of dash and is measured using. So contact angle, you may have heard about this contact angle measurement. So the answers for this question of a part is that contact angle on the surface is indicative of hydrophilicity, and it is measured by osmometer. Second is contact angle on the surface is indicative of osmolality, and it is measured by osmometer. Third is contact angle on the surface is indicative of hydrophilicity and is measured by goniometer. Another fourth answer is osmolarity and goniometer. So the answer of this question is that contact angle on the surface is indicative of hydrophilicity. By using the contact angle measure, this technique or contact angle measurement technique, the hydrophilicity or hydrophobicity of the sample is measured. And the instrument which measures this contact angle is called a goniometer. So in the next slide, I will explain in a detail. Like in the upper picture, you are seeing that there are different types of surfaces: superhydrophilic surface, hydrophobic surface, and superhydrophobic surface. So super means highly hydrophobic, and hydrophobic means normally hydrophobic. So as you know that this contact angle measurement is done using the water. So with the help of the water dispenser or syringe, water droplet is placed on the sample. After placing the water droplet, if the surface is hydrophilic or superhydrophilic, the water it will disperse on the surface as the surface is water loving hydrophilic. And if the surface is hydrophobic, it is water hating, the water droplet will remain intact as a droplet and it will not disperse as the surface is water hating hydrophobic. And this hydrophilic leads to the wetting of the surface, and hydrophobic, as the surface is hydrophobic, it will lead to non-wetting because hydrophobic surface will not be wetted by the water. So by this technique, the hydrophilicity and hydrophobicity of the surface is measured, or hydrophilicity or hydrophobicity of the material is also measured as the water is placed in the material surface. So how is it measured when this droplet is placed? Like here I am showing the super hydrophilicity case. The droplet completely disperses and it will form the sheet-like structure, and the angle between the surface and this water droplet is very less. It's maybe equal to zero if it is wholly absorbed by this hydrophilic material. And if it is hydrophilic but not that much hydrophilic, the water droplet will be here, but it will disperse from some extent, but not fully. So the angle between this line, the upper line of the water droplet and the surface is called the contact angle, and this contact angle is measured. And more the contact angle, for example, greater than 90, 120, 150. More the contact angle, more is the hydrophobicity, sorry, and less the contact angle, less is the hydrophobicity, and more is the hydrophilicity. So using the contact angle measurements, you can see the hydrophilicity or hydrophobicity of the surface. And in the questions, hydrophilicity was mentioned. So another, now talking about the instrument. So why is a very simple instrument where there is a light source and and uh, and um, and in the front of the light source there is a water dispenser and sample is placed on the stage, and after this water dispenser places a drop of water on the sample, and this there is a camera which captures the image, and the image which is captured is connected to the computer, and the software it analyzes when the droplet is placed on the surface. After taking the image, the, the, cam- the software analyzes this angle between the surface and the water droplet. And this instrument is called a goniometer, and it is used to study the contact angle or surface wettability of the material. I hope you have understood this. Now I will move to the next question. Next question is concanavalin. Concanavalin A is a the answer is carbohydrate binding protein, protein binding carbohydrate, protein binding lipid, and D part is lipid binding carbohydrate. Actually, this concavalin A, it's a protein and it binds to the carbohydrate. Thus, it is a carbohydrate binding protein. Actually, this concalavarian is a lecithin, lecithin, which is a carbohydrate binding protein, and it was originally extracted from jackpea, which is called Anabila ciferum. And this concalavarian A, it is found to bind to the various carbohydrates like sugars, glycoproteins, and glycolipids. And the binding of this concalavarian is irreversible binding. And this concalavarian, it is used to in the cell culture. It is used to activate T cells or natural killer cells. So the answer for this question is concatenary is a carbohydrate binding protein. So then, now moving to the next question, the question number 8. The question is retina contains rod and cone cells. Rods detect dash and cones detect dash of the light. A answer is intensity. Rods detect intensity and cones also detect intensity. Second is rods detect color and cones also detect intensity. Third answer is color and color. Fourth answer is intensity and color. So what is the answer? It's a very basic question. You must have studied it in 11th or 12th class, maybe in the 10th class also. So the answer is D. Rods detect intensity and cones detect color. So in the retina of the eye, there are two types of cells which are electrically active cells or photosensitive cells. When these two cells are rod cells and cone cells. So these rod cells, they are placed in the outer segment uh, and contains the rhodopsin, which is the photosensitive pigment which is present in the rod cells, and it is a visual purple in color. These cone cells, they are outer segment. Their outer segment is conical, and they contain the pigment, photosensitive pigment named iodopsin. These rod cells, their inner end has a small knob, and cone cells, their inner end is branched. These rod cells, they are sensitive to dim light, and they are usually used to visualize in the dim light. And these cone cells, they are sensitive to bright light or and give daylight photopic vision. So these cone cells, they are activated in the day when there is more light, and they are used they are used uh, they are used in the daylight or when the light is very bright. These rod cells do not, as these rod cells contain the rhodopsin, and rhodopsin doesn't have the capability to detect the color. So these rod cells do not give the color vision. It does not help to give the color. So the cone cells are three types. There are different types of these cone cells. We detect the blue, green, and red color. So they help to visualize the color. Insufficient rhodopsin results in night blindness, as this rhodopsin helps to see in the night light or dim light. So it leads to night blindness. When there is less light, the person will not be able to see. Insufficient iodopsin results in the color blindness, as the cone cells contain the iodopsin, and if there is a mutation, which leads to the insufficient iodine. It results in the color blindness. You may also aware that some persons are blind to some particular colors. That is because of the insufficiency of the iodine in the particular three types of cells, for example, blue, green or red if these cells are not present, if one of these cells are not present. So these rod cells, they are far more numerous, they are present in more numbers as compared to cone cells, that is cone cells of sugar. Now moving to the next question, which is also related to the rod and cone cells. The question is, rods and cones are electrically active, sound sensors, pressure sensors, electrically inactive. The answer is very simple, that these rods and cones, they are electrically active because this light signal is converted into electrical signal, and then it is and it this signal is analyzed by the rods and cones. So these rods and cones, they are also called photoreceptors, and they uh, they are the nerve cells which make the retina, which are usually in the part of the retina. And there are also other cells like glial cells and different other types of cells in the retina. But mainly the photosensitive cells are rods and cone cells. And it is known that this retina contains approximately 125 million rod cells and 67 million cone cells. We already said in the earlier uh, slide that rod cells outnumber the cone cells. So the last question is, which is question number 10. The powdered collagenase should be stored at all times, desiccated at but at what temperature this powdered collagenase should be stored? Answer is plus 20, 0, minus 4 or minus 20. So what is the desiccated collagen? Desiccated collagen is actually a desiccated powdered collagen is which does not contain any water, which is the desiccated. Uh, which does not contain this water, or water is removed completely from this. So this collagen is you may be aware of this collagenase. Collagenase is a proteolytic enzyme that breaks the bond, the peptide bond between the collagen and disrupts the ECM or destroys the ECM. And this collagenase is stored at minus 20 degree. And in a powdered form, it's also stored at minus 10, 20 degree. But if you, it is advisable that when you dissolve the collagenase after dissolving, it should not be stored for a long term. So if it is stored, it is stored at minus 20 degrees centigrade, so it should be in a desiccated or desiccated form or powdered form. So you can store it for a long term. But if it is in a dissolved state, it is only stable for 1 year in a dissolved state, and that too at minus 20 degrees centigrade. So this was all about the week 4 session of this course. I will see you in the next meeting, or next live session, which is in the coming Tuesday, 7 p.m. So thank you for your attention, and thank you for joining.